Yes. I'd like to emphasize that, in fact, we are currently in two concurrent pandemics. The cholera pandemic began in the 1960s and continues to this day. And COVID-19, of course, we all know because we've been suffering through it for the last 3 or 4 years. We're going to emphasize the waterborne diseases because it provides a really useful mechanism to demonstrate the very powerful interaction of the environment on human health, and it's going to be exacerbated with climate change. Clearly, it's obvious when there's about 1.5 billion cases of diarrheal disease, including cholera, and close to 2 million deaths each year, that waterborne diseases, when you take into account all the other aspects of health associated with safe water, it becomes clearly a very important human consideration. Cholera is a global disease. It's a water-related diarrheal disease. And as I said, the seventh pandemic began in the 1960s and continues to this day. It occurs in about 50 countries. It affects at least 7 million people every year. Unfairly, the Bengal Delta was known as the native homeland of cholera outbreaks. However, 100 to 150 years ago, cholera was rampant in the United States, in Canada, in Europe, and in other countries of Asia. It's because the cholera bacteria exist in the aquatic habitats, and with the evidence of new serotypes that have occurred in the last decade or so, it's clear that we can never eradicate cholera, but we can control it. Now, what I'd like to emphasize is a disease that is simply characterized in the laboratory. The bacterium can be easily grown in the laboratory, and the disease is devastating in the loss of fluid, but with replenishment of fluid, victims can be saved. The work that we have been doing goes back some 40 or 50 years, and interestingly, the bulk of the basic research was done in the Chesapeake Bay, which is on the east coast of the United States. We did most of our studies going back to the 60s, the 1960s, along the various stations that we still continue to sample even today. The discovery early on was that the vector of cholera is a plankton component, the copepod. Now, there are many other creatures in the aquatic environment that can carry the bacterium, but in fact, it's the natural host in the copepod, which has as its commensal and perhaps even symbiotic flora the vibrios in general and Vibrio cholerae in particular. But these other factors of weather, rainfall, salinity of the water, temperature, movement of animal species carrying the bacterium, all of this plays into what we now deal with as an understanding of the environment for which this bacterium is associated. This model was developed some 22 years ago, and earlier. Actually, the work began in 1975 in Bangladesh. We developed this simple model from the work in the Chesapeake Bay, namely that in warm spring months with increased water temperature and sunlight, phytoplankton, which carries chlorophyll, become very abundant. The zooplankton feed on the phytoplankton, and when the zooplankton population ceases to be dominant, then the breakup of the zooplankton releases into the water column the vibrios, and without water treatment, drinking the water, as was done 100 years ago in the U.S., transmission of cholera occurred. However, what's very interesting is that the criticism for our model was that, well, perhaps that exists in the Chesapeake Bay, but does it really fit the events in Bangladesh? So with that challenge in 1975, we began our work in Bangladesh. Indeed, we were able to find that bacterium is present in a variety of living creatures in the environment, in the aquatic environment of Bangladesh. But again, the copepod turns out to be the major culprit. Clearly, person-to-person transmission is very important. And ICDDRB, including Dr. Kadri and colleagues there, have done magnificent work showing this relationship. However, the transmission is clearly important, but the source is the environment. And that we have spent a good deal of time working on. More recently, we've taken some of the isolates from various parts of Bangladesh and have been able to show that there is an increase in antibiotic resistance of the Vibrio cholerae from the environment. And this seems to be mirroring what's been happening with, in fact, patients coming to the hospital. Now, it was very early on that it occurred to me that with this aquatic relationship, back in 1985, which was before many of the students in this audience were born, the launching of satellite sensors, that satellites were launched carrying sensors that could measure chlorophyll, sea surface temperature, and sea surface height. So 25 years ago, we put together the capacity to use satellite sensing for predicting when cholera would become a threat. And it was a very successful output because 22 years ago we published this paper in the Proceedings of the National Academy of Sciences, showing, in this case, the blue line is sea surface temperature, and the red line are the cases of cholera just off the coast of Bangladesh in the Sundarbans region in the Bay of Bengal. Now, this was very crudely done because at the time all we had was Landsat, the satellite that measured chlorophyll, sea surface temperature, and sea surface height. And what we did was very crudely measure these parameters in a cubic, a mile dimension just off the coast of Bangladesh in the Bay of Bengal. It was a crude relationship, but surprisingly coincidental and accurate. We've been able to improve the model to understand that cholera actually occurs in 3 different modes. The endemic mode is when throughout the year along the coast in the Bay of Bengal and along the coast of India, there will be low levels of cholera and then spikes in the spring and fall, concurrent with the blooms of plankton populations. Whereas inland, as in Delhi, India, or in Chhattisgarh in Bangladesh, epidemic cholera occurs, which is sporadic, and the outbreaks are usually quite deadly. And it occurs after disasters. And then in Dhaka City, there's a mixed mode of cholera, 1 or 2 seasonal peaks, and then continuing throughout the year. We've since improved our prediction capacity significantly. The model now is much more sophisticated. But the fundamentals are rainfall, water temperature, insufficient infrastructure or damaged infrastructure from social chaos or from gathering of festivals of various types. And with the launching of many, many new satellites, starting very early on in the 1960s with the Vibrio hypothesis, the 1980s using Landsat and then CWIF, another satellite that was launched with sensors, and then our publication in collaboration with the team at the National Aeronautics and Space Administration, NASA, we were able to publish the first study. Subsequently, we now have been able to use other satellites and sensors, and that allowed us to identify the mixed modes of cholera that I just mentioned. And then more recently, we're now using highly sophisticated satellites that have been released to the public, including some of the previously classified sensor satellites that we now can use to monitor movements of population. And that allows us to bring socioeconomic factors into the calculation of risk of cholera outbreaks. In 2017, we tested our model by doing a retrospective analysis of cholera in Yemen. As we all know, that country has suffered civil strife and the worst cholera epidemic in history, really up to that point. We did our retrospective analysis with our predictive model, and then the actual cases, as you can see, showed that the model back then was at least 60% accurate, and since we've been able to move it even to 80 or 90%. In fact, in the last few months, in May of this year, we have been surveying globally hotspots, potential hotspots, and to our great interest and concern, we showed that in Mariupol, Ukraine, there is a risk which is modest at the moment. We detected that risk in May. It has been enhanced by continuing observation in June, and our model strongly suggests that there will be a cholera outbreak, and in fact, it's very interesting, some cases have begun to occur. So this is an ongoing predictive capacity, and we are now working closely with the United Nations agencies and the British Aid Agency to introduce public health measures for that country that's undergoing all sorts of strife, including the invasion. I'd like now to speak about the genomics component, because that's really very important. And in fact, that's what this conference is all about. Genomics, all of the omics, as Dr. Cowdery pointed out beautifully, and engineering, bioengineering, and genetic engineering, bioinformatics. Our technique, developed back in 2007, is to extract the DNA, which is now pretty standard for many research workers, and to be able to sequence, regardless of what the sequencer used, that is, it's agnostic. That allows us then to match the sequence reads with an extraordinary database that's close to 200,000 curated samples, about 160,000 for sure, but we're adding curated genomes on a daily basis. And this allows us to identify the microorganisms, the bacteria, the viruses, the fungus, and the protists, but even more importantly, to identify the presence of genes for antibiotic resistance, for pathogenicity properties, for metabolic activities. So that means microbial identification and pathogen characterization. So now we are well launched into the microbiome of the environment. Now, it's important to identify the species and to be able to do identification really down to the strain. That is more important, and even substrain, variants, mutations, etc. And it's important to identify antibiotic resistance and epidemiological tracking. Now, there are some very elegant techniques that are currently available. Mass spectrometry, the Maldatov, allows accurate identification of antibiotic resistance, and then the various PCR tests, and other than the whole genome sequencing, subtyping is important. And obviously, this provides analysis of phenotypes and antibiograms, and we can do surveillance. But with whole genome sequencing, as I've just described, you can do the entire panoply with that analysis. And so this provides elegant strain subtyping, which these other techniques cannot do, at least for the entire community. And it allows us then to be able to know the pathogen and the community of pathogens, and then to be able to respond appropriately. Now, whole genome sequencing, particularly in the clinical laboratory, has, starting in 2007, has really increased significantly, but it's pretty much, for the moment, restricted to the wealthy countries, the US, the UK, South Africa, but it is now being carried into a global capacity, and it will transform medicine. Let me give you one example. This is a study we carried out, which needs to be written up. I'm a bit slow getting this into publication. Carried out with the Cholera and Enteric Diseases Institute in Calcutta, in Iceland. They provided us with sets of samples over a period of time, phase 1, 2, and 3, where they knew by classical techniques that specific pathogens, namely Vibrio cholerae, because these are patients coming into the cholera hospital, and then samples where they have difficulty identifying the causative agent, and then a set of samples as well from individuals who had no symptoms of diarrheal disease or certainly none of cholera. And the findings were fascinating because with the known disease etiology samples, that is, they would send us the DNA extracted and blinded, while they did the classical techniques for viruses, bacteria, protists, etc. And here, the pink area represents pathogens. We were able to pick up Vibrio cholerae, but we found that we picked up not just Vibrio cholerae, but other pathogens, perhaps Campylobacter, enteropathogenic E. coli, Salmonella, Shigella. And so the point I'd like to make is that these infections are polymicrobial. Koch's postulates have been very effective for 100 plus years, but we are now in the 21st century with the omics revolution, and it is time to understand the community structure and the polymicrobial interaction of pathogens that cause disease. In the case of where the diagnosis could not be made by classical culturing, etc., we were able to determine that indeed dominant was the various pathogenic E. coli that comprised the pathogenic invasive factors. And then in the healthy individuals, it turns out that a significant carriage of pathogens without symptoms of infection. We downloaded the Human Microbiome Program database from the NIH and found that indeed Westerners carry pathogens, the pink area, but not at the extent as in the Calcutta samples. Interestingly, the probiotic, represented by blue, is larger, a fraction for the healthy population in Calcutta, and not so dominant for the Western population. And this is quite likely related to diet, in that probiotic, yogurt, fermented milk, etc., is a staple of the diet in India. A little perturbing was the fact that antibiotic resistance amongst the samples where identification was difficult, except by whole genome sequencing, and in the case of the known samples, but also the healthy individuals, a spectrum of resistance quite strong to the beta-lactams, the macrolides, etc. And so this, I think, is a function of the accessibility to antibiotics without prescription in India. And one of the results, by going back and looking at the actual isolates, we found that indeed there's reduced susceptibility to two important antibiotics, the ciprofloxacin and the azithromycin. And so this then, I think, as part of what we all now recognize as a global third pandemic of antibiotic resistance. Now let me show what we've been doing very interestingly. Right now, a continuing assessment of Vibrios in the Chesapeake Bay. And this is the work of Kyle Brumfield, graduate student, my colleague, my wonderful colleague, Dr. Buck, and others in the laboratory, where we are now developing a predictive model for Vibrios other than Vibrio cholerae, because in the Chesapeake Bay, we do have a problem with Vibrio parahemolyticus associated with food poisoning, and Vibrio vulnificus, which is very serious. It's not in a pandemic form, that is, there are not thousands of cases. There may be a dozen cases. However, the fatality rate is about 50%. So it becomes important to know when and where these other Vibrio species, pathogenic to humans, can be predicted, which times of the year, and geographically where we would find them. We've begun to analyze our data, and this is a study of the antibiotic resistance profiles of these. And you can see the red depicts the resistant incidence, 90% almost, resistant to ampicillin and penicillin, and some resistance, some of the other antibiotics, in the crop of Vibrio parahemolyticus isolates from the Chesapeake Bay. A little more concerning is not such a high percentage, but a more general resistance to antibiotics for Vibrio vulnificus, which, as I've just mentioned, has a high fatality rate. So this means that an infection that can occur, let's say by an open wound, someone recreationally swimming in the bay, or a waterman who is harvesting oysters and crabs and cuts a finger if it gets infected, that individual must go to the emergency room and be treated, but its antibiotic resistance profile is absolutely critical. Now let me switch to COVID-19. And you, of course, will say, well, what has this virus got to do with the bacterium Vibrio cholerae? It turns out COVID-19, and I won't go into the symptoms, we all know most tragically what they are, and we really have suffered a terrible 2.5 years, losing friends and family and acquaintances to this disease. And it has a long COVID with the neurological symptoms seem to persist, and loss of smell and taste, etc. But very early on, back in March and April 2020, when COVID-19 was just beginning to be recognized as a pandemic, we tested our capacity and found clearly from respiratory samples we could detect the virus and subsequently its variants. And this has proven to be a very powerful tool using the genomics approach that I have just described. Others, very early in 2020, showed in a number of studies, as you can see here, a dozen or so, that in the stool, the virus could be picked up, significant numbers. Furthermore, in this particular study, which was early in 2020, two years ago, in the 41 patients, top to bottom, and horizontally, the red line indicates for that patient the throat swab was positive, and the yellow orange line indicates the stool was positive. So you can see that the stool remains positive in most individuals for quite a long time, a month or two, after the throat swab or the nasal swab has gone negative. This is a very important feature of this organism. In addition, as I will mention a little later, airborne transmission is critical. Now, we started work with the government of Maryland, the state government, the governor, and the Department of Environment of the state of Maryland, and the Department of Health, collaborated with our team. And we sampled, as you can see, some intensely populated areas around Baltimore, and wastewater treatment plants with high population, and some remote areas, not so remote in the same sense as from Bangladesh, but outlier areas that were not in dense populated cities. We used not just the one PCR gene as the tracker, but 2 or 3 genes. And of course, by studying wastewater, this is an accumulation of domestic as well as industrial discharges, and the search then in the wastewater for the community understanding of COVID-19 allowed us then a powerful tool. And here's some data very early on in 2020, June. We began our work through the auspices of the state of Maryland, working collaboratively with a wonderful team in the Department of Health and Environment. And the 2 lines show the blue line is the target gene recommended by CDC, the Centers for Disease Control in the U.S. And the orange line represents the additional genes we used to be employed to ensure that we were really picking up COVID-19. And as you can see, in late July 2020, 2 years ago, we began to detect the presence and, sure enough, cases in Frederick, Maryland, where this particular analysis was done, began to appear. Now, one interesting study, just as a good example of how this technique is powerful, is that we began analyzing the wastewater samples from specific dormitories at one university. And by testing the specific dormitory wastewater, tapping into it at the effluent at the dormitory, we found in one dormitory presence of COVID in the wastewater. And immediately, 221 students in that dormitory were tested. Interestingly, 10 of those students were found to be positive, but 9 were asymptomatic. It could have been a hugely serious spreading event, but it was able to be nicked in time because that dormitory, those students, were quarantined for the 2-week period, and that averted then the spread throughout the campus. Now, we have subsequently been very intrigued with the metagenomics, all of the population of microorganisms in wastewater to determine if we can pick up association of other pathogens, bacterial and viral and fungal and protists such as Giardia and Cryptosporidium, any relationship with the COVID, either in the intensity of the infection, the longevity of it, whether it has a relationship to whether it goes into long COVID. And so this paper has just been published this week showing the metagenomics, the microbiome profile of wastewater. This was published by MBio, one of the major journals of the American Society for Microbiology. And indeed, we are beginning to see some of these relationships of phyla, genera, species, and the community structure of a COVID community. We have begun analysis as well to characterize the expression of antibiotic resistance in the wastewater. And you can see over a period of time, the variation in antibiotic resistance, which can be very interesting. We have completed and published another study with the California Los Angeles Water District, where they were reusing wastewater from the sewage plant with proper treatment, of course, because of the drought in California. So the Q1 water is the input from the sewage plant. The sludge goes out to sea. And we were able to show, by working closely with the Orange County Water District, that after for filtration, biofilm removal, and after reverse osmosis, the reduction in antibiotic resistance was significant. Now, why is this important? Well, the concern was by mixing wastewater with lots of antibiotic resistance genes being carried by the wastewater bacteria with clean water in order to make available more water to the community, would there be transfer of antibiotic resistance to the naturally occurring bacteria? And the answer is no, because the treatment processes remove the antibiotic resistance factors. Let me now put all this together. In collaboration with Dr. Jutla, former postdoc with whom I have worked the last 10 years and continue to be associated, brilliant young man and his team, we now provide prediction in association again with the same prediction hub at the University of Maryland, working together to be able to provide them a capacity to know during the year when Vibrios are dominant, where they're dominant, and to be able to send out alerts to the public. We have been able to convert the model developed for cholera that I've just described to the SARS COVID-2 virus, because there's a distinct relationship with a variety of factors that we can measure again by satellite. Various temperature and movement of populations, the structure or the capacity of the environment to be able to understand whether or not there is a resilience to being able to bring together the public health needs, and then the socioeconomic factors of dense population areas, the wastewater analyses and so forth, bringing all this together. The hypothesis for environmental COVID-19 risk prediction is based on its very powerful aerosol transmission capability related to humidity and humidity factors, some of which can be measured by satellite, and the movement of populations, again measurable by satellite. Because it turns out that in the comfort zone for us humans, the temperature is somewhere between 17 and 24 degrees Celsius in the US. And when it's cold, then there's a tendency, of course, for people to go where it's warm, or in the ambient environment, the built-in environment, and then the potential for infection goes up. Similarly, when the outside temperature is very hot, the tendency is for humans to aggregate where air conditioning, hence the built environment, is cooler, and the tendency and the risk of COVID goes up. So, putting all these factors together, we tested this conversion of the cholera model to the COVID model way back in April, May of 2020, 2 years ago. And this was our risk map, and this was the COVID case map. And for a first trial, it was really very significant. And here is the environmental risk map for a year later, where the concentration of the number of cases has gone down, but nevertheless we're still able to detect the risk and then show the actual cases. And so we're getting close to some 80% accuracy, which is quite remarkable. And we provide now, on a regular basis, if you go to this particular website, you will be able to get the COVID risk county by county for the United States. And we're moving this to global prediction. We've just been funded by the NASA $1 million funding program to be able to launch this on a global basis. I want to acknowledge colleagues from the ICBDRB and wonderful group of people. I'll shout out for Dr. Alam and certainly Dr. Kadri and all of the team there, the NICED team, and many students at the University of Maryland. And then let me just give you one last very exciting work that Kyle Brumfield is the first author, and there's a whole team of us together. We've put together, we've correlated